Hello, everyone. Welcome to the April edition of MycoTalks. My name is Runhui Liu, and I'm at Weill Cornell Medicine in New York. And my co-chair today is Dr. Alexandra Brandt from University of Exeter. We have two excellent speakers today for you, Runhui Liu from East China University and Alastruey from Instituto Salud Carlos III in Spain. The philosophy of the MycoTalks is diversity on career stage, gender, geography, field. Each talk is 30 minutes, and we have after that Q&A. We go in depth discussion, so please put your questions in the Q&A banner here, and we will lead the discussion there with Alex. There are upcoming talks in May and June. The MycoTalks in May will be by Linhui Wang from Chinese Academy of Science and Heather Conti from the University of Toledo. I also would like to introduce to you the upcoming MycoTalk clinics, which is another edition of the MycoTalks, focus on clinical studies, case studies, and experts in that area. So the next MycoClinics will be on June 2025. So with that, I would like to switch to my co-chair, Alexandra Brandt, who will introduce the first speaker.
Okay, thank you very much, Lilian. Well, today it's my great pleasure to introduce the first speaker, Professor Runhui Liu from the East China University of Science and Technology. So Runhui earned his PhD from the Department of Chemistry at Purdue University in 2009. He then undertook postdoc research at the California Institute of Technology and the University of Wisconsin-Madison. But at the end of 2014, he went back to China and started his independent research career in the schools of materials science and engineering at the East China University of Science and Technology. And that's in Shanghai, and I believe that's where he's speaking to us from today. And he's won several awards for his outstanding work. His research encompasses the area surrounding biomaterials, antimicrobials, and anti-cancer agents in the context of tissue damage and tissue engineering applications. Today, he's going to talk to us about his amazing work on peptide mimicking amino acid polymers and the next generation of bioactive materials, including host defense peptides and antimicrobials. So welcome, Runhui, and we really look forward to hearing your talk.
Thank you, Alex. Thank you very much for your kind introduction. I really want to take this opportunity to express my great thanks to the kind invitation. Give me this great opportunity to share our work in the past 10 years. Let me share my presentation. Okay, so suppose you can see my presentation now. So
yes,
the
that's
topic,
great. We can see it. Thank you.
Okay, thank you very much. So so today I want to share the topics, part of the work in our group about host defense peptide mimicking antifungal studies. As we all know that microbial infection has been a great challenge to human health, and ever since the first antibiotics has been used in clinical applications, all these drugs used in practice will have these resistant problems because microbes will have multiple mechanisms or different ways to develop resistance against these drugs. Compared to the bacterial infections or drugs, these fungal infections for a long period of time has been relatively ignored because the fungal infections normally will affect these immune damaged people, compromised people, but people with normal health condition, and that most of the cases is not a huge problem. However, this change ever since the COVID pandemic, and we can know that the fungal infection has been increasing, both for their the number of the patients and also the cost, and as has been recorded even for only the Canada albicans. So each year, has been large amount of people have the life-threatening infections, and we have to pay attention to that because these kind of like the system infection has very high mortality rate. A part of that, the reason is because there are very limited drugs available compared even to the antibacterial drugs. The problem is because fungi and mammalian cell are both eukaryotes, they're very similar. So it's extremely difficult to develop potent antifungal drugs, but still with low toxicity, or we call the good selectivity. In addition, there are more and more drug-resistant fungi emerged in recent years. So this has been the serious problem for the whole human being. So in our study, we have been very interested in the host defense peptide, HDP, because this kind of peptide exists in almost all forms of life, including us, human beings. They have very interesting structure. They have the positive charge and amphiphilic structure. So their mechanism is different from conventional antibiotics. They will target the cell membrane. This is the general mechanism. Because like the microbes, they are outer membrane, we have the negative charge lipid. So the positive charge whole defense peptide can interact with that, and they have the amphiphilic structure to disrupt the membrane. So this mechanism brings the advantage of HDP because microbes is hard to develop resistance, and also this HDP can have broad spectrum activity. However, this natural HDP also have their innate disadvantages. First, they are very easily to be hydrolyzed because their structure is not stable, especially in the physiological condition. Another thing is those peptides are very expensive. If we want to make the large quantity of those peptides, normally we need to use the solid phase synthesis. So they have been very time consuming and expensive. What we can do? So from literature, there has been a large amount, over 1,000 different types of host defense peptide has been isolated. They have very high diversity in their structure. They have alpha helix, beta sheet, hairpin turn. But very interestingly, even they have the diversified structure, they have very common characteristics in their structure. They have positive charges, now 2 to 9 positive charges. They have over 30% of the hydrophobic residues. That means the amphiphilic structure. So those two give us very important starting point for our research. They have the structure diversity. That means it's possible for us to optimize and even modify the structure of HDP. And they have the common structure characteristics that tell us it's possible for us to mimic their structure to mimic HDP. So in the past 10 years, when I get independent in our group in China, so we try to mimic host defense peptide to mimic their amphiphilic structure under the positive charge. These are the common structure characteristics. And first, we want to address their problem of natural peptides of their unstable structure. So we introduce a natural amino acid by introducing different identity of the amino acid, different combination, different ratio, and different polymer lengths, and also the terminal functionality. We can introduce a large library of the peptide mimicking compounds. So we can tune their stability against proteolysis. We can make that during using the polymerization methodology. This is one part polymerization, so can make those compounds very easily in large quantity. Also, the price is also low. When we try to mimic the peptide, normally the structure of the backbone to make peptide is very limited. One is the alpha amino acid polymer. Another one is to make the peptide or the beta peptide. The latter two is try to have this unnatural backbone to resist proteolysis, make the structure more stable. However, realize in many of the cases during our exploration, we either have the compound, especially for antifungal study, we either have a compound not active again, or we have the compound very active but also very toxic, or they are active, also they are very low toxic, however they have favored the in vivo performance, like the pharmacokinetic and others. This tells us if we can have any new structure to mimic peptide, they give us a great opportunity. And then we figure out whether we can transfer the carbonyl from the main backbone to the side chain. Then we get to oxazoline. This is a very old backbone. They have been used as a polymer. They have very hydrophilic properties because our group is only methyl and ethyl, no other functional group. So we think about if we have our group with functionality like the amine, like the guanidine and others, so probably we can use polyoxazoline to mimic as a new backbone to mimic peptides. So we do that and we demonstrate that works. So when we use those polyoxazoline to mimic the peptide, we can quickly find the one have potent activity against methicillin resist as always, the bacteria. We have selective and potent activity. And when we try to find potent and selective activity against the fungi, we failed, even we synthesized maybe over 1,000 of the compounds. Because as I mentioned previously, for fungi to have the selective antifungal activity is much more difficult. So one thing we think about, because there are some research mentioned that for the fungi, their organelle can be, they take this bacteria and they evolved to get what they have now. And some literature have already told that the organelle membrane is very similar to the membrane of the bacteria. Then they give us a new idea, because our group also work on the drug delivery, the cell-penetrating peptide. We think about things we cannot get a selective activity against fungi bacteria, fungi membrane directly. Can we combine the host defense peptide and the cell-penetrating peptide and make the compound penetrate into the cell, fungal cell membrane, and getting inside the cell to target the organelle membrane, because organelle membrane is more similar to the bacterial membrane as has been reported in literature. Okay, take all this together. So we design the library of polyoxazolines with the backbone of the host defense peptide mimicking with amphiphilic structure, and we introduce polyarginine because this is the very characteristic functional group of cell-penetrating peptides. And we are very lucky for the very first site of a compound, we get potent activity against both C. albicans and C. neoformine. So the strain we have here are drug-resistant strains, and many of them are isolated clinically, the pathogenic strains. We can see the MIC minimum inhibitor concentration of our compound against C. albicans will be around 0.78. It's even higher, it's even better than amphotericin B. This is a potent antifungal agent. Fluconazole is not active because these are all drug-resistant strains. Similar for C. neoformine, our compound is even a little active more than the amphotericin B and even much better than fluconazole. More interestingly, because our compound is have excellent solubility in water, And after testing B, we know that the compound itself doesn't dissolve well in water, and it also have high toxicity, but our compound have low toxicity, low hemolysis. So that means it has good selectivity. Another very important question is, if we continue to use those compound, whether these fungi can develop resistance. So we do the test. So we keep using our compound on the fungi, both for CLBK and Cneomyces, and over generation and generation, see what happened. And we found that none of these fungi can develop resistance. That's what we really want to see. When we design these kind of antifungal compounds, we combine the host defense peptide and the cell-penetrating peptide. We propose the compound will get into the fungi first. But is that the real mechanism? To test that, we conjugate fluorophore to our compound, and then we co-culture our compound with propylene iodide, PI, and the cell became the cell together. Well known that PI doesn't penetrate the cell membrane. When we see the fluorescence of the red color, that means the cell died, the membrane damaged. Then the PI can get into the cell to have the red fluorescence signals. So from the time-lapse confocal, it's very clear that we can see the green fluorophore get into the cell first. So that means our compound get into the fungal cells first. And over sometimes something happened within the cell. The membrane damaged, the cell died, and we see the PI signal. So this is something very similar to what we propose for the mechanism. And it's the same thing for C. neoformans. In addition to confocal, we also test the TEM. So the left panel will be the normal cell for CLBCAN and C. neoformin in TEM, very smooth membrane. And after treatment with our peptide mimicking compound, we can see the concaved the membrane. However, we don't see obvious damage or the hole-like damage. Something only like that concave structure. This is something very like the cell-penetrating peptide, because the cell-penetrating peptide can get into the cell membrane and have the transient small damage, and then this can be recovered by the cell itself. And when we go to the TEM, the left panel is the normal TEM of CLBCAN and the C. neoformin. After treatment, we can see the disruption, obvious damage of the organelles inside. And what happened? And we care the most about the cell nucleus and the mitochondria. That's the two very important fungal organelles. So we have the stain with the nucleus as the blue, the staining at the central of the cell, and the red spreaded spot, the very characteristic staining of the mitochondria. After co-culture with our compound, we can see both the blue staining of the nucleus and the red this staining of the mitochondria will diffuse into the whole cells. So that means we break the membrane of the cell nucleus and the mitochondria. These are the two target, the organelle target of our compound. And for C. neoformin, we observe the very similar phenomena. After we figure out the mechanism, we want to know how the performance in vivo of the compound. The first, we make the murine skin infection model, and we compare our compound with the antifungal drug nystatin. So the results show that our compound is pretty active to very significantly reduce their drug, the fungal loading. And also we can see from this the slide of the tissue, without treatment, there are plenty of this fungal invasion into the tissue. After treatment of our compound, most of this the fungi are cleaned from the tissue. And also our compound has no dermal toxicity. That's pretty safe. And we also test another model, the murine fungal keratitis model. So in this model, we compare our compound with amphotericin B, the very potent the antifungal drugs. We can see that the performance of our compound is comparable to amphotericin B to significantly reduce the fungal loading. And for the eyeball, we can see after treatment of our compound amphotericin B, the eyeball is more clear compared to the non-treated group. So for the vehicle group, we can see a large amount of this fungi hyphae invaded into the tissue. After treatment of our compound, we can see most of that is cleaned out. And also we showed our compound has no corneal toxicity. So how about the systemic infections? Because that in the clinical practice will have a high mortality rate. Then we establish the model and see if the mice after the systemic infection of C. albicans without treatment, the mice will die. All the mice died very quickly. After we do the treatment with either the control amphotericin B or our compound, most of the case we can save the mice. And whether that's the systemic infection effect, then we dissect the mice and track this fungal density in different organs like the heart, liver, spleen, lungs, and kidney. For all these organs, we can see our compound can reduce this fungal loading as effective as the amphotericin B control. So that tells this is a real systemic antifungal effect. So this is a very characteristic tissue slide of the kidney. Without treatment, we can see large amount of this fungi invasion into the tissue of the organ. And then after treatment of either amphotericin B or our compound, most of these fungi are cleaned out from the organ, from the tissue. Whether our compound is biologically safety, and we inject the compound from the tailwind of the mice, and then over different time point draw the blood and test the blood biochemical parameters. And we can see that for all different time point, different time window, the blood chemical parameters are all within the normal range. So that tells our compound is pretty safe without the acute toxicity. As I mentioned before, the mimicking host defense peptide, the very important part is to mimic the cationic and amphiphilic structure. So we tune the linker or the arm length of the side chain from 3 carbon to 4 carbon, 5 carbon to increase the hydrophobicity and decrease that to 2 carbon and even 1 carbon methylene to reduce the hydrophobicity. Using that, we can tune the hydrophobicity and amphiphilicity of the whole compound. And interestingly, we can see that the shortest linker with only the methylene as the arm has the highest antifungal activity. The MSE against the C. albicans and the cineoformin could be as low as 0.1 or to 0.025. For those kind of peptide-like structures, it is pretty much out of our expectation. It's important to mention that this is not only fungal static because it also the fungicidal. We can see this is a MSE in the practice is a MBC. So the MSE and the MBC value is almost identical. More interestingly, this kind of compound potentially can penetrate the blood-brain barrier, the BBB, to use for the treat of the fungal meningitis infections, because this is a very tough target. First, we want to see in vitro whether they can penetrate the BBB. So we get these brain blood endothelial cells to get this monolayer on the transwell. This is a very classical model. and we make sure that cover the whole area to check whether there's no the cell between there, if there's no the gap between the cells. So we check their the electric resistance. The high resistance tells there's no gap between the cells. And then we put our compound into the well, but we do see that over 0 to 24 hours, our compound can pass out this monolayer to get into the lower solution. That means our compound potentially can penetrate the BBB. Encouraged by that, we want to see how our compound can distribute in different organs. So we inject our compound into the 10-way and tailwind, and then see the distribution. Of course, you can see the liver and the kidney have very high the distribution. This is normal and not strange because those two are the metabolic the organs. But it's very interesting to see in the brain, we see much higher the level of the distribution of a compound than other organs like the heart, the spleen, and the lung. So that tells us our compound can penetrate BBB and get into the brain. We also use another model to double check the result. This zebrafish model, because this is OT zone, the brain, the very black, the control cells, our background is clean, and the fluorescence dye itself doesn't penetrate the BBB. So the dye doesn't penetrate. We can see almost no fluorophore signals. And after the dye conjugate with our compound will bring that into the OT zone, the brain. We can see very strong fluorescent signals here. They also tells our compound can penetrate the BBB, get into the brain. Encouraged by this, we established the mitogenesis model for both CLPK and cineophormin and do the system administration of a compound. We can see for either CLPK and cineophormin, our compound performance is comparable to F-tyrosine B to reduce the fungi loading in the brain. This is our recent paper in the JACS. So this is the brain tissue slide without treating all the saline control group. You can see a large amount of the fungal 5-alpha invasion inside. After treatment with F-tyrosine B and our compound, most of these fungi are cleaned out from the brain. We also do the system toxicity study to inject our compound from the tailwind and draw the blood and see all the parameters are within the normal range. That means this compound is also pretty safe. Pretty much that's the last slide. This is our recent work about our design of the dual targeting antifungals. As I mentioned at the very beginning, to find the antifungal compound with potent activity and low toxicity, all the good selectivity is extremely difficult. During this finding of the drug resistance for either bacteria or cancer, because all the drugs, either bacteria or cancer or fungi, can develop resistance. Many researchers think about whether we can either use multiple drugs together or we can design in one drug molecules with different targeting ability. That means we use one molecule to target work on two targets. In that case, they can significantly reduce the possibility of drug resistance. So this kind of study have a successful case in either antibacterial and anti-tumor studies, but for antifungal it's extremely difficult because even for single target designing of antifungal drugs is very hard. I have to say that we are pretty lucky because we have been focusing on to mimic the amphiphilic structure to target the cell membrane. So in this case, we designed the polyisozoline with the positive charge and amphiphilic structure. we want to target the cell membrane. And of course, we can get this selective anti-fungal activity by targeting the membrane. And by that time, we think about whether we can also target the DNA. So we think about we can introduce some of the very simple structure like the naphthalene. It's known that the naphthalene can get into the DNA during using the peptide stacking. We also check other different structures, and this very simple naphthalene structure already work. So in this case, we use the naphthalene structure to have the peptide direct interaction with DNA as one target, and we have the positive charge amphiphilic structure to target the fungal membrane. So this one, we get the real dual targeting antifungals, but also with good selectivity and potent activity. Because all this performance either in vivo and in vitro demonstration is pretty similar to what I introduced before. So I don't want to go to all the details for this animal study or other activity studies. So pretty much my time is there. I will stop here, and I will finally acknowledge all my group members and the funding from all this agent. And thank you all for your patience, and I would like to take any questions you may have later. Thank you.
Thank you, Runhui, for this presentation. So we will have the questions after that. So now we are moving to our next speaker.
Okay, thank you.
This is Dr. Alastruey. So she's a senior scientist at the Mycology Reference Laboratory of Spain. Her primary research interests include public health mycology, diagnostics, epidemiology, and antifungal resistance. And in this capacity, she chaired the technical expert group that was responsible for the development of the first fungal priority list, the infamous one which was published by World Health Organization in 2022. So she's a president also of the Spanish Society of Mycology and representing Spain on the European Confederation of Medical Mycology Council. So she has other international activities. She's a chair of the fungal infection study group, and she has also directed the Latin American programs, and she's a fellow of both ESCMA-ID and ECMM. So with that, we'll go to Anna's presentation, and we are looking forward to it.
Thank you very much. Thanks for the nice introduction, and also for inviting me to be here today. Can you now see my presentation, hopefully? Yes?
Yes, looks good.
Perfect. So yeah, thanks again. I'm gonna switch to a completely different topic, which is raising awareness in public health. As already been said, I've been involved in the last years in several of the documents that I will comment today. So I just wanted to bring you with an overview and update of what is it. Just to start with, I think we all know this, but I think it's good to set the scene that we know that fungal infections are mostly unrecognized, but they have supposed a burden of an important amount of burden of disease in the world. And importantly, they have been linked with high mortality rates. Even with susceptible strains, we are talking about rates of 30, 40%, but they can be reached 80 or 100% in some diseases. In addition, recently, we've been witnessing an expanding in geographic ranges in endemic pathogens that were supposed to be restricted to some regions, and now we are seeing how they are expanding, as well as emerging of new pathogens, as we know for Candida auris, for example, that is mainly resistant in it raised in the last years, and this has also bring a lot of attention to fungal infections. In addition to Candida auris, we can also include here Trichophyton indottiniae, which is also a dermatophyte causing superficial infections that is resistant to different antifungals. In addition to this, and talking about the resistant, we've seen that the use of fungicides in agriculture, at least we can say that selects for antifungal resistance in the environment, and this can be then cause infections in humans. So just to summarize this set the scene, as I said, there is a higher numbers of invasive fungal infections, especially because there is rising immunocompromised populations that are expanding globally. We have more and more hosts susceptible for fungal infections, as we learned from COVID, but also patients treated with different biological treatments that are at risk for fungal infections. As we said, the mortality and morbidity of fungal infections are high, and we only have 4 classes of systemic antifungal drugs. And they are expanding geographical niches of some endemic mycosis that are sometimes primary pathogens able to cause infection in immunocompetent population, and there's also an emergence of antifungal resistance. And also, it's very important to highlight that the global access to antifungals and to the effective and affordable diagnostics is completely unevenly distributed along the world, and it's not especially available in places in which fungal infections are very frequent. So with this problem in mind, WHO started to bring together several initiatives in which I have the privilege of and honor to participate in most of them. It started, I remember the first meeting we had in ECMI 2018 in Madrid, when we started a group of people to discuss about the inclusion of Candida in the GLASS program of WHO. We will talk about later, but definitely what it really started, I think, all this movement was the development of the fungal priority pathogen list in 2022, that I guess all of you have not only read, but seen hundreds of times in slides, reports, manuscripts, and grants proposals. Since 2022, a lot has happened. In 2023, the 2 documents were published, the research agendas in Human Health and in One Health, and I will introduce a little bit about this later. In 2024, GLASS decided to include already last year Candida in the surveillance system, and is now being included there. And also in the EFSA, the European Food Safety Authority, has been for several months assessing the impact of azole fungicides in agriculture, and I will also discuss this. This was published in January, but all the work in December and January last year. And finally, like 2 weeks ago, 1st of April, WHO had released 2 important documents that they reviewed the antifungal pipelines and diagnostic landscape. So my talk is going to be more or less about all these documents, trying to summarize what has been done in the public health in the last years, and also where are we now. So as I said, I start with the fungal priority pathogen list, and very briefly, because I guess most of you have already been heard about this and read it, but it was a big effort of a lot of people with several steps and looking at a lot of literature and resources. I always have to say that whenever I present this, people always ask why this pathogen is not included here. And I have to say that the list was really limited at the beginning. It was only 3 or 4 species. And we acknowledge that the problem was bigger and more species were included. And you can see here the 19 species that were selected at the beginning, and then the 10 criteria among which these species were prioritized later on. Take into account that we perform like systematic reviews of all these 19 pathogens for all the criteria that we defined, and we also ran 2 surveys among the global people working in mycology, and also some experts in public health, to try to come up with a final list that you all know, and it's divides the pathogens in these 3 groups. One of the main things of the FPPL was like there's a lot of knowledge gaps, and this is part of our our aim to try to provide more data in order to be able to better prioritize these pathogens, because a lot of things we could not prioritize due to the criteria, because there was a lack of solid data to classify this. I'm talking about complication and sequela, or mortality in many cases, burden of disease, trends, some information that is really lacking there. In addition, in the priority list, we also highlight 3 priority areas for action: public health interventions, research and development, and surveillance. And there are several actions described there, but I will discuss a little bit further this later on. In 2023, and after the publication of the fungal priority pathogen list, 2 research agendas were published by WHO. One was the Global Research Agenda for Antimicrobial Resistance in Human Health, and the other one from the quadripartite in the One Health Priority Research Agenda for Antimicrobial Resistance. In this last one, the One Health Agenda, it's true that specifically fungi are not that specifically described throughout the document, but in the glossary you can say that they are specifically and clearly state that antimicrobials include fungicides and antimicrobial resistance includes fungi, which is a big thing because most of the people think about antimicrobials being equal to antibiotics. So this is highlighted in this publication already, showing the importance of fungal infection. But in the Global Research Agenda for Antimicrobial Resistance in Human Health, in which I also have the honor to be part of the expert panel, several points among the 33 priorities were specifically targeting fungal infections. And these are 2 in diagnostics. You can see that mainly it's about the priority fungi, like critical priority, Candida auris, Aspergillus fumigatus, Candida albicans, and Cryptococcus neoformans. Investigate and evaluate point-of-care diagnostics tests, and also the clinical utility of the diagnostic accuracy of phenotypic antifungal susceptibility testing. In the treatment and care, investigate antifungal regimens, optimize for efficacy, cost, safety, and duration to the treatment of infections caused by the priority pathogens, especially including that in setting with increasing or high prevalence of antifungal resistance. And these, apart to these 3, another 3 were included in the cross-cutting theme to investigate the prevalence, incidence, morbidity, mortality, and socioeconomic impact. And this is very important because it's necessary to really prioritize those pathogens. And then it's specifically highlighting across geographical and socioeconomic settings in population experiencing vulnerability. Also investigate the association, contribution, and impact of structural and health system factors and people with comorbidities, and identify optimal surveillance methods to generate accurate and reliable data. These are really important because the data is necessary to really inform the rest of the processes. So apart from the FPPL, these 2 documents, agendas, priority research agendas, were published in 2023, in which these fungal infections were also highlighted. In 2022, after 2019, there was the publication of the early implementation protocol for Candida in glass, and this was a group of people that put together with WHO and like a pilot study about if it's feasible to implement Candida into the glass candidemia. So it was from bloodstream infections caused by Candida in the glass platform, and it has officially been included in 2024. You can see here in this graph where AMR surveillance in invasive fungal infections is included here with AMR surveillance and let me see, can you, oops, wait, sorry. If I can hear that, yeah. So you can see here AMR surveillance in Candida bloodstream infections was included into the glass platform. Here you can see all the surveillance activities. This is within the glass web page as well, and you can see here how glass fungi is highlighted here. You can click, and there is this specific work package of glass fungi in which AMBAR surveillance of fungal infection is included. So starting last year, glass participants are able to include candidemia cases in the glass survey system. You can see here all the participating countries, and there are differences between, so there are some countries that contribute only to antimicrobial resistance, some countries that contribute to antimicrobial consumption, and some countries that contribute to both. But you can see that most of the world is represented in this program, and despite having limitation, of course, because there are differences in how the resistance is screened and what the denominators, at least we could have a good amount of data regarding antifungal resistance in Candida in the next years. In addition to this, as I said, a very important initiative was carried out by EFSA, the European Food Safety Authority, along 2023 and 24, in which they want to evaluate the impact of the use of azole fungicides in the development of azole-resistant Aspergillus, and it was an effort commissioned by the European Commission, and it was coordinated with EFSA, but involved other 5 different agencies: the European CDC, the European Chemical Agency, European Environmental Agency, and European Medicine Agency. And all this, I won't go into much detail here, but you can see that there are a lot of working packages. Each agency was responsible of different, and there was a big coordination, and a lot of people participated here, of course. What the outcomes, you can read the full document in the EFSA webpage, but the outcomes and the implication, they show that between 2010 and 2021, approximately 120,000 tons of azoles were sold in the EU area for non-medical purposes. And we have to take into account that not all the countries provide the same amount of data. And most of these were used for plant protection with a stable annual average of 10,000 tons. And there is enough and substantial evidence that supports this link that we've been advocating for, accept between the environmental azole fungicide exposure to the selection of Aspergillus and the cross-resistant to medical azoles, what is called the environmental resistant route, and the link has been demonstrated for Aspergillus fumigatus, of course, because that's where we have more information, but it's less clear for other Aspergillus. The non-medical use of azoles contributes to the environmental selection of azole-resistant Aspergillus fumigatus, potentially leading to infections caused by azole-resistant Aspergillus fumigatus, but more research is needed. Based on ecological selection dynamic, identified several hotspots, both in agriculture and in biocides. And the prevalence, human azole resistant, we know it's really variable among populations and also among different geographical regions, but has significant clinical implications with high mortality rates, but some other infections, as we know, CPA or AVP, are less documented. So the key recommendations of these documents is that to combat azole-resistant Aspergillus, is recommended to have a comprehensive strategy in all these fields: agriculture, medicine, research, biocide use, and EU approval of fungicides. And as I said, I think the whole document is very interesting, and there are a lot of annexes that you can read through the document in the EFSA website. And finally, as I said, 2 weeks ago, 1st of April 2025, Also, a couple of documents have been released from WHO, the antifungal agents in clinical and preclinical development. That has been the first analysis from WHO. It covers only systemic antifungal drugs in development. So as the same as the FPPL, it doesn't cover superficial infections, and this is a limitation, as we know that there are a lot of fungal infections. Most of the burden is in superficial fungal infections, but they are focusing on the ones that have higher mortality rates and impact in survival. And it critically evaluates the activity and the innovation potential of these candidates to address the infection caused by the priority pathogens. So it's, again, a very nice document that I recommend you to read if you are interested in this field. Here you can see how the different antifungals have been discovered and developed, and you can see how many years does it cost from the discovery to the approval of different antifungals. For example, here in amphotericin B, it was discovered in 1953, and the first approval was in 1997. And we know we still use that antifungal, but imagine, so there's a lot of years that it costs to develop a new antifungal. In addition, here you can see another interesting table in which they include all the priority pathogens here, and here the antifungals in the pipeline, and they see if they are active in green, possibly active or not active, and also if there is this information in base of peer review data or not, and if they include in vivo or only in vitro data. So this is also very interesting. Here again, the number of products that target each of the priority pathogens here. Of course, as you can see, most of the new agents are covering the critical priority pathogens. This is also important, or it's straightforward, because of course these are the ones that are associated with higher burden, the ones that have more numbers, and then that's why most of the developers try to target those species. In addition to the document, there is a dashboard that you can also consult online in this page, in which you can see different ways of representing these pathogens. For example, here again, the number of the different antifungals in the different clinic fungal pathogens, and if they are active, not, or here the indications, if they are label extensions or novel clinical agents, or the stage of development, phase 1, 2, 3, or if they are approved by clinical product type, or here where they look at the new agents by mode of action. So this is a very interesting review in which they highlight that in the past 10 years, only 4 new antifungal drugs have been approved, and only one class is approved every decade. From the analysis, it has been clear it's 1 per decade, and pediatric indications and optimal pediatric formulations are really rarely available. And 9 agents are currently clinical development. 4 of them meet at least one of the innovation criteria of the WHO. 7 agents show in vitro and in vivo efficacy against at least 1 critical pathogens. As we saw before, most of them target the critical pathogens, although they also target others. And the WHO Global Review of Antifungal Agents captured 22 products there, but the main take-home message is the pipeline remains extremely sparse and insufficient to address the global need to prevent and treat drug-resistant fungal infection. In addition to this document, the same day, and it has been an effort of a lot of people as well, as all these documents, the landscape analysis of commercially available and pipeline in vitro diagnostics of fungal priority pathogens was also released, and this is the first landscape analysis of these diagnostics. the main result is that there is a fair number of tests available, but most have several of these limitations, including long turnaround times, limited scope of pathogens that they cover. They relied on the fancy equipment or well-equipped laboratories, on very well-trained staff. They sometimes have inadequate sensitivity or specificity, and they are not well suited to primary and secondary health facilities. This document also includes a lot of tables and graphs, and you can see here how the different platforms are analyzed here, providing specimen, microorganism, sensitivity, specificity, advantages and disadvantages. The final goal of this document was not to produce a catalog, although it looks like a catalog. There are many things that have not been captured there, and I know this is one of the main limitations that I have been highlighting since the very beginning to the team, that there is so much, there is not a common source in which you can find all these diagnostics, so you can always leave things behind, and this has happened, of course. But it's mainly trying to look for the main gaps, and that's the aim of the document, to really review and see where are the gaps and what we need to work on. And they also include commercial assays for antifungal susceptibility testing, which is more and more important due to the rising antifungal resistance. We know that there are some available for yeast, but the ones for molds that are validated and well suited for antifungal susceptibility testing in molds are more scarce. In addition, there are also the characteristics of different platforms in different annexes, and you can see also examples of these here. So I recommend you to see them, to have a look at them if you want, if you are interested. So the gaps and needs detected there is that there's a limited knowledge of the impact of antifungal resistance in LMICs. It has been seen that there is really almost no commercially available test in LMICs. There are fewer commercially available automated tests for identification and lack of board regulatory approval for identification, and for filamentous fungi, and inadequate sensitivity and specificity of most tests. There's a limited availability of non-culture-based methods. Here we not only talk about PCR, but also antigens and antibodies and so forth. So beyond Candida fumigatus, Aspergillus fumigatus, Cryptococcus neoformans, and Pneumocystis jirovecii, the number of tests is very limited outside of these four. There are not many multiplex text platforms suitable for use in level 2 facilities, so they are mainly suited for tertiary hospitals, which we know is where most of the severe fungal infections are seen, but it would be advisable to have more that could be accessible and available in level 2 laboratories, and especially to test antifungal susceptibility in an appropriate time. And also there is a few lateral flows to identify invasive fungal disease, and not available multiples for use at any level of the health system. So the research priorities in these documents is that we need a simplified blood culture platform for BSL-2 laboratories, also antifungal susceptibility testing automated for yeast and filamentous fungi, multiplex diagnostics to identify fungal platforms, and if possible also resistance, and improve LFAs. So with this, I have more or less reviewed all the, from my point of view, most important international agencies document that had been delivered in the last few years. So as conclusions, the public health awareness, I think, has raised in the last years with the publication of these documents, but all the documents highlight that there are big knowledge gaps in the field and lack of data. We need better diagnostics, and we need to make them accessible globally. There are a limited number of antifungals in the pipeline, and this could be a big problem because the antifungal resistant rates are really rising, and also the global access of these antifungals is really a big limitations. In many countries don't have access to most of these of antifungals that have been in the market for more than 20 years. Surveillance systems are starting to include fungi, but they're still far from perfect, and many need to be developed and implemented. And there is a real need for education and awareness, and all these documents highlight this gap, and not only within the scientific community, general public, and patients. And then there is a One Health approach needed, but it's a bit complex. So just to finalize, what's next? What are we working on now? As we said, many people is asking about how to include more priority fungi in the list, and we know that species like Sporotrix or others Aspergillus are probably important enough to be included. So now we are working in the global blueprint for the implementation of the fungal priority pathogen list, which main objective is to outline actionable interventions to really move the needle in the global response efforts to fight fungal infections, and engaging global, regional, and local sectors to create partnerships, which is key, advocate for sharing resources, data, and knowledge, and also stress the importance of increased investment. So we've been working a lot in this blueprint along the last year and a half. We had the face-to-face meeting in Istanbul in October. And if you want to know more about this, you need to go to either ISHAM in May or TEAM in September in Bilbao, because that's where it hopefully will be presented. And with this, I'm finalizing with a big thank you to everyone. Most of you, or many of you, are already in the chat or have contributed. It's impossible to acknowledge everyone here, and just highlighting some of them, especially Hatim Sati, that has been the project leader of the FPPL and the blueprint from WHO. Valeria Gigante has been the lead for the pipelines, both the antifungal and the diagnostics. Jorge Mateu, the One Health agenda. Silvia Bertagnolo in the human health agenda. Daniel Marcano has been with Carmen Pessoa and Silvia also in the GLASS document, and all the scientific community with, you can see, recognize many faces here, and it's impossible to name everyone. The global mycology societies and the financial support. And I think we have to acknowledge also that the biggest, probably the biggest public health awareness thing that we have had is the Last of Us, that is really creating at least the question if a fungal infection is going to be a threat in the coming years. And then with this, I would like to thank you for your attention, and really happy to take any questions.
Great. Thank you very much, Anna. And now I can welcome everybody back to the screen. Interesting news. Diagnostics is still not terribly high on the success list at the moment. So interesting talk. Okay, so Runhui, are you there? Yes.
Yeah, I'm available.
Yeah, so I'm going to kick off with questions for you, and I remind everyone who's listening today, if you would like to post a question, please put the name of the person to whom you're addressing the question. Okay, so I'm going to kick off with a couple of questions here. So the first one is, would you please tell us if this host defense peptide mimic is currently being developed as an antifungal drug by any pharmaceutical company at the moment?
Okay, that's a very critical and important question. In fact, there are multiple pharmaceutical companies. They are trying to push forward on this road. Multiple drug candidates are so far already in clinical trial, either in phase 1 to phase 3. and it's possible in the near future we can see those kind of drugs available on the market. But you know that everyone know that the drug development will be a long like a time and very kind of like we see it is kind of like risky. But we see that there's a great opportunity. We can see those, and more of these candidates probably will go to the pipeline.
Right. And sort of as a second part of that question is, how stable are the drugs that you, in the format that you've developed them? So particularly thinking about with reference to the cost of supplying and storing them in low and middle income countries.
Yeah, this another very important question. So regarding the safety, I think there are two different way to think about. One is their stability against the proteolysis, like the in vivo, and another stability is something like whether that's stable on the shelf, like at the room temperature or even the high temperature, like the sunlight. When we develop these kind of host-defined peptide mimicking compounds, we try to introduce the unnatural amino acid. So in that case, we can tune the stability. Part of the work in our lab I didn't have time to introduce today is that we develop a new synthetic methodology to make those polypeptide or peptide mimics. This new methodology enable us to synthesize either kind of like alpha amino acid or their analogs or even the backbone like polyoxazoline. Those are totally stable against the proteolysis, and they are also very stable on the shelf. So some of the compound, for example, we can have those the peptide mimic compound on the shelf at the room temperature for years, and or even the solution still keep their original activity. But for us, because different application either for like drug development or the new antimicrobial biomaterials, different case we have different kind of like requirement on that stability. So for scientific research, our work has pay attention to this, so we can use our synthetic methodology to prepare or design the backbone with that is totally biodegradable or totally not degradable, or we can tune like between the degradable and non-degradable. So it depends on the application requirement. So far, I think we are able to do that.
Okay, all right. Thank you very much. I'll pass over to Ilian now.
Yeah, so the next question is to Anna. So people are very interested how the updates of this fungal priority list will be happening. For example, there is a question here from a participant in South America asking if Sporotrix will be included, but we can imagine that other fungal species will come up or resistance of some species that were not resistant before. So what will be the perfect process here? How that list will be updated? What will be the timelines? How would you coordinate with the World Health Organizations or with other entities?
Yeah, thanks for the question. I think this has been so. In the first FPPL, it was already discussed that the idea would be to update this every 5 to 7 years, and actually this was after. So the bacterial priority pathogen list was released, and the new update has came up last year. So in this blueprint document that we are developing this year, there's going to be this need of starting to update the list, and we are currently starting to work on that already. How this works is mainly there are two phases. One is in the pathogens that need to be included in the list, and this needs to be, the case needs to be made to include this pathogen in the list. Since 2022, many people have been discussing about why some pathogens are not there, and so we already have some key players, as we discussed, Sporotrix or other Aspergillus, because only Fumigatus is included. But the main important thing is that we need to produce data. So why Aspergillus flavus was not included in the 2022 fungal priority pathogen list because there was not enough data justifying the burden of disease and the importance in human health, take into account this comes from the human side. So these two sides, like really generating the case and on the other side producing this data. And also we are getting a lot of requests, well, WHO is getting a lot of requests to update the list. And this has been included in the original document as well, that it has been, the procedure has to be adapted to different regional situations, because we know that they are geographically distributed pathogens, that here in Spain, we don't have many histoplasma cases, but in Guatemala, they have more histo than TB in people with HIV. So there are a lot of requests from different partners to try to adapt the list to the situation, and that's also a part of the process.
Okay, thank you.
Okay, so Runhui, I've got two questions linked really to what's the dual purpose peptide mimics. So the first is, was the vacuole membrane integrity monitored in addition to mitochondria and nuclear membranes? And what is the timing of the effect? Which of those two were disrupted first?
Oh, okay. Well, very interesting questions. So during our study, as I mentioned during the talk, I think the two very important of this organelles is mitochondria and the nuclear. And like the vacuole membrane, so for the TEM, like we can see, at least for the sineoforming, clearly we don't see obvious damage, as I can see from the TEM. So we didn't pay very close attention to that. Another reason is that the target we monitored very carefully. Those are the main target. Normally, we have a kind of like a lot of the cascade reactions once they are targeted. So so for our so far, we think that the main target are those two. The white cloud, the membrane, is not something so far we can see obvious damage within the short time, because the window we have is like within minutes. That's something that can happen kind of like quick. So you can see from the time lapse, sometimes when the peptide mimicking compound get into the fungal cells, it will happen to have the damage or the cell death during the very short windows. So the time of the interaction is very quick. So the main organs that will have a big impact on the cells will be the main target, as we figure it could be. But it's possible for different structures or different compounds that may can also target the vacuole membrane. But so far, I have no idea whether that can be faster than that can target the mitochondria or the nuclear membrane to disrupt those organs.
Right. So
Yeah.
a kind of link to that is another question about, so it's, what is your strategy for developing these HDP mimics for use in other fungi? Do you predict that each will be fungus specific? And with the dual function peptides, could you speculate whether you might find that one function, for example, the membrane targeting function, will be pathogen specific, for example, but the intracellular component might be usable across a range of fungi, for example?
Okay. I think this is a good and also a tough question, because when we try to design the antifungals, first the select antifungals is extremely difficult to design and to finally get those one with potent and selective activity. Otherwise, there will be not short of antifungal drugs available on the market. And this raises a really very interesting and difficult question for us, because I also have this kind of questions for not only for antifungal, but also for antibacterial development, drug development from the expert in hospital, whether we can specifically target some strains but not on others. this is possible for bacteria. We already have this kind of result. For fungi, once in a while, we can see the compound that they have potent activity, for example, maybe from against the C. albicans, but not so as potent as Aspergillus. But this kind of like not surprising because Aspergillus normally is more tough than Candida. So I don't want to put this kind of like the design the selectivity. So the good thing probably will be something that they can target Aspergillus, but it doesn't really affect Candida. That can be interesting. We do have one of the case is that, for example, when we have the like the intestinal tract system, we have this benign the kind of like the fungi, like we have this some of the fungi that's kind of like benign to our body. So whether we can differentiate those kind of fungi from the pathogenic fungi. One of the compound from our pack, we didn't publish that yet because we want to have the application collaboration with the industry. We can differentiate those two. Those doesn't target the benign fungi, but kill the pathogenic fungi. This is very interesting. Both are fungi, but this very specific case is very hard to generalize this kind of like a conclusion.
Yeah, that's an interesting one because we don't really understand the role of commensal fungi yet in microbiomes, and if you could avoid targeting them and leave them alone while targeting pathogens, I think that might be important in the future when we have a better understanding of microbiome function.
Yeah, because of that kind of application, they want us whether we can add our compound into yoga, and because that can, it doesn't hurt those kind of like the germ inside yoga, but it can pass the stomach and also probably have their functions in the body to kill the pathogens.
Yeah, interesting, very interesting. Okay, over to you, Elian.
So Anna, there is a question about the environmental drivers of Aspergillus triazole resistance. So how about heat pollution and any of those, environmental drivers that are coming with the climate change and with the pollution and all that?
Yeah, thanks for the question. That's like a very interesting route, and there's a big hypothesis, and I think it has mainly started by Arturo Casadevall, that said that climate change is like really making more fungi pathogenic, and there's this confirmation about Candida auris that apparently it can be the global warming making it pushing some species to become more thermotolerant, because you know that Arturo also had this theory that our body temperature of 37 degrees is just because that's the optimal, the best cost-effective temperature to get rid of the fungal infections, not being susceptible to fungal infections. So this global warming, it's an hypothesis that is probably pushing some fungi to be able to grow at higher temperatures and then being able to be pathogenic and as Candida auris. So I think that's an important and interesting and probably good argument for that. And also the pollution, there has been several studies in which it has been described that more polluted air can contain more Aspergillus fumigatus. So that's also another thing. And of course, the use of fungicides in agriculture, I think, is more than proven that it's at least, if not generating, at least selecting azole resistant. And that's like, I think we should not question that anymore and just move forward and try to avoid this dual use or try to mitigate the impact of this dual use of antifungals, as we have a couple of new fungicides with the same mechanism of action as antifungals for medical use that are in clinical trials.
Yeah. Thank you.
Okay. Runhui, I've got a couple of questions here with your because you're disturbing the plasma membrane in order to get the functional other function of the peptide inside the cell, do you think that your peptides would work in a combinatorial way to instead of being a sort of dual purpose, could it actually work better with a combinatorial approach?
Yeah. This is a very good question. In fact, we have been our lab has been working on this for quite a while. Although we haven't published the work, but this is a kind of like something very I think useful and practical strategy. We have tested our host-defined peptide mimicking compound to have a combination with other different antifungal drugs and other even those kind of like the new compound in literature all from the herbs. Many of those we do see like not only just the combination, but we see quite some of them have the strong synergism on that. We have one of the paper is now reviewed in Nature Microbiology, and we just received the feedback from the reviewer. I think it's pretty positive. Those kind of work will be something we already see the kind of like a combination of synergism. And I hope very quickly we can address all those kind of like questions from reviewer and they can be published and everyone can see that. So I have to say that this is also a strategy not only developed by the pharmaceuticals. It has been studied in our labs. It has also been used in the clinical applications. This is something, this kind of host-defense peptide mimic compound can work and probably can have like a different way for combination for different purpose. I think there's a lot of opportunity we can find from this kind of strategy. Thank you.
Okay, good. So here's another question. How would you compare your approach to the development of non-peptide host defense molecule mimics? And then there's a citation of Larwood et al. Regarding antifungal activity of brillocidin. Are you able to comment on that, or is it a bit short notice to answer that question?
Okay, I'm not quite sure I catch the question. So what compare our peptide mimicking?
Yeah, to the development of non-peptide host defense molecule mimics.
Non-peptide host defense mimicking.
Yeah.
Okay. I think the backbones will have impact on their the outcome of their activity and the biosafety and the in vivo pharmacokinetics and a lot of things. For just the mimicking the compound, ever seen the pioneer work from like a Professor Samuel Gelman, they used the amphiphilic to mimic use a polymer without the secondary structure to mimic that. We know that this is a good strategy. So now I think the focus of the point is which kind of backbone is relatively easy, always more rationale to develop as like potential drugs if like evaluated by the pharmaceutical companies. Most of the case, I guess they will prefer something that will be closer or similar to our peptide or proteins because they're kind of like kind of drugs, including the antibodies, whatever peptide drugs can already has been the majority of the new drug development. So I think it depends on how they can easily accept this kind of like a strategy to develop new antibiotics. For activity and others, I cannot say which backbone is better because when we work on the polyoxazoline, this is something like not a peptide backbone, just like the mimic backbone. We can see some of the special case, their activity and the performance in the body is even better than the peptide, all the alpha amino acid peptide. But when we think about try to push forward for like a real drug development, still depends on how the big pharmaceuticals will think whether this is very kind of like more acceptable by our traditional thought on this drug development.
Yeah, yeah, okay. Ilian, back to you.
Yeah. So we are actually reaching the end of this. So there are 6 minutes, and I will ask 2 questions here to Anna myself, and I think we'll wrap up after that for interest of time. So Anna, you said that the collection of more data on emerging pathogens is very critical. So can you elaborate how that collection is done and who's going to collect that data? What data are you looking for, and who's going to collect it?
Yeah, that's a very important question. So, as I said, we developed 10 criteria for ranking the pathogens for the FPPL. And this criteria is mainly, as we said, is fungal infections in the human health. So it's like we need more data about burden of disease, mortality, inpatient care, disabilities, treatability. So we have a lot of data for candidemia, for Aspergillus fumigatus, fair data, but for most of the other pathogens, we really are lacking. So, and the problem here, or not the problem, I think it's an opportunity for us as a community, is that we really need to join forces, because one of the limitations is that we don't really have very good diagnostics tests. And the ones that we have, they are not available globally. We know that fungal infections happen mostly in areas of tropical and subtropical regions where many low and middle income countries are. So they have no access to diagnostics or limited, also less to antifungals. So we really need to make an effort to try to get all these diagnostics to as much as countries as possible, to develop ones that could be suitable for them, and then as a community gather enough data. Mainly the clinical people really need to investigate on the cases and really gather databases. And it's not only important for fungal infection for the FPPL, I think also for this analysis of big data to be included in all these AI systems. We really need to provide solid data to the public, so it's better understood the burden of disease, how these infections develop, and how to tackle them. So I know it's not easy. It's a big,
No, it's
big
not. Yes.
task, but I think that's the beauty of our field, that it's going to develop in the next years, and we have a lot to do, and that we have a very nice community that could make a lot of impact in this.
Okay, thank you. And the last question that I have, I think it's more outlook, which is probably good for closing here. So you're working on this interface between the science, which you communicate with your working groups to local governments, to governments, and you're hoping that science will have actually impact on policy and all that. And there are 2 successful stories, I would say. The World Health Organization recognition of the fungal pathogen, this is one of them. And Last of Us is another one that brings actually people thinking more about fungi in a more unconventional way. So can you kind of wrap up with another successful story or another success that you're hoping that will be coming from those efforts?
Yeah, I think, or at least my impression has been that more and more founders are including fungal infections into their calls. So we see more and more budget being available for fungal proposals. And I also see more governments sensitizing to fungal infections and requesting data about that. So I think you named the 2 most really impactful things. And I have to say that despite being very involved in the FPPL and all the WHO work, most of the interviews I got from the journalists in my country have been when The Last of Us was launched. So we really need to acknowledge that the general public need to be more close to what we do. And I think that would make to have a bigger impact in the policy, and hopefully our ultimate aim, which is really help the people that suffer the fungal infections.
Okay, thank you. So with that, we'll be closing the session, and thanks a lot to our speakers. And I'm really glad that this actually is still ongoing, the MycoTalks, and there are many people that are attending, and there are a lot of views on YouTube, which is fantastic. And thanks to Alex for co-chairing with me. It's been always pleasure. It's probably second or third time that we are co-chairing together. And with that, yeah, we are hoping to see you next month.
Okay,
you,
thank
everybody.
you.
Bye now.
Yeah,
Bye,
see
thank
you.
you.
Yeah, thank you. Bye.